Good afternoon, everybody, and welcome back to my channel. My name is Dakota. I'm a first-year medical student at the University of Central Florida College of Medicine. I'm going to be covering a variety of topics on this channel that range from biochemistry all the way through anatomy and my second-year curriculum. I'm kind of doing this as a way for me to review for step one, but also to help other students like yourselves master the material and things like that. So with that being said, let's go ahead and start on today's lesson, which is collagen synthesis and its related pathologies. So before we talk about the main steps in collagen synthesis, we need to kind of discuss some of the different types of collagen as well as its properties. So collagen is one of the major proteins in the human body. And just for numbers' sake, it composes about 30% of all total protein in the body. And it's found almost exclusively in animals. And some of the major functions of collagen include, its role as connective tissue, its ability to provide high tensile strength, specifically within joints and bones. And collagen is very resistant to stretching, and we can kind of look at the structure and see why. But before we do that, let's go into the 4 types of collagen. So the first type of collagen is type 1 collagen, which makes up mostly bone. And an easy way to remember that is the word 1 is located in bone. So type 1 forms bone. Type 2 forms cartilage. Another way to remember that is you can spell cartilage, cartilage. The third type of collagen makes up the arterial walls. And it comprises a couple other things, but the main thing here is the arterial walls. And we'll see what defects in type 3 collagen look like when we talk about Ehlers-Danlos syndrome. And the final type of collagen is type 4 collagen, and this makes up the basement membrane of cells or tissues. Okay, so types 1 through 4, there's many other types of collagen, but for the sake of the step 1 exam and what we need to know for class, these are the 4 main types. So now that we've talked about the different types of collagen, let's kind of go into the properties of collagen itself. And so some of the properties of collagen include the fact that it is a long asymmetric molecule, and its composition is roughly 33% glycine, 33% and 30% of either hydroxylated proline or hydroxylated lysine. And then one of the other key characteristics of collagen is that it has this repeating tripeptide sequence. So within the primary structure of collagen, we see this repeating tripeptide sequence, which consists of glycine, followed by an X amino acid and a Y amino acid. And most often, this X is proline. And if you remember proline's amino acid structure, it has that R group that forms a ring with the amino group of the backbone. And so it's not one of the traditional amino acids in the sense that the R group comes off and it's free hanging, not attached to anything else. Proline is actually attached to the backbone of its own amino acid. And after that, the Y is often hydroxyproline. And this X and Y can be switched out for lysine as well, and the Y would then be hydroxylysine, but most often we see proline and hydroxyproline in this primary sequence of collagen. So now that we've talked about the properties of collagen, we need to look at some of the post-translational modifications that occur during the synthesis of collagen. So we can get hydroxylation, which is essentially the addition of OH groups. And specifically, we add these OH groups to proline or lysine side chains. We can also get glycosylation, which is the addition of a disaccharide. Sorry for the terrible handwriting. We can get addition of a disaccharide specifically to a hydroxylysine residue. We also get proteolysis of our procollagen chain as it's secreted out into the extracellular matrix. And then we can also get covalent cross-linking, because in order to form this collagen molecule, it's essentially the covalent cross-linking of a lot of tropocollagen molecules stacked on top of each other. So we'll look at the mechanism of how that happens when we talk more about desynthesis. So now that we talked about the different attributes of collagen and the properties that it has, let's go ahead and take a look at the actual synthesis pathway. So synthesis of collagen takes place in a specialized cell type called fibroblasts. So let's go ahead and talk more about how it's synthesized. So to set up the kind of scenery, we have the nucleus here. So the nucleus, that's where transcription occurs. And so we need to transcribe an mRNA transcript that's going to encode for these pre-procollagen proteins, these alpha chains, okay? So what happens is the nucleus transcribes these alpha chain transcripts. The mRNA is secreted outside of the nucleus through nuclear pores, and it's translated in the cytoplasm by free ribosomes. As the ribosomes start to create that polypeptide, it gets sent into the rough endoplasmic reticulum. And for those that don't know what the rough endoplasmic reticulum is it's basically a site for a lot of post-translational modifications that occur to proteins. So in this specific case, our pre-procollagen is going to get shipped into the rough endoplasmic reticulum where it's hydroxylated. And so this pre-procollagen is going to get hydroxylated by an enzyme called lysine or proline hydroxylase. Okay? And it's lysine or proline depending on which amino acid is hydroxylated. So scientists aren't very, clever when they come up with enzyme names. So this enzyme is called hydroxylase. And what does it do? It hydroxylates amino acid residues. Okay? This enzyme needs two main things to function in the fibroblast. It needs vitamin C, and it also needs iron. What we'll see in some disease pathologies is that oftentimes these enzymes are non-functional because of a variety of things. In this case, there's a disease that we'll talk about called scurvy that results in a vitamin C deficiency. And we'll talk more about the effects that has in future slides. Once these proline and lysine residues have been hydroxylated, we can also get a process called glycosylation to occur. And what glycosylation is it's the addition of sugar moieties or disaccharides onto these amino acid residues. And specifically, glycosylation occurs on hydroxylysine residues. So we'll attach our sugars in the endoplasmic reticulum, and that's just another form of post-translational modification. After hydroxylation and glycosylation have occurred, we now have our pre-made pre-procollagens, which are essentially just alpha chains of polypeptides ready to go. And what forms the procollagen monomer is essentially the aggregation of three alpha chains. So we're going to take three of these pre-procollagen chains, and we're going to essentially create hydrogen, and disulfide bonds between the chains. And they're going to create this triple helix. And that's what we call procollagen. So it's ready to be exported out of the cell into the extracellular matrix, and we'll see how that process occurs. So now that we've formed these hydrogen and disulfide bonds on the ends of the procollagen, it's going to actually get packed up into a vesicle, transported through the Golgi, and exocytosed outside of the fibroblast. And when it's outside of the fibroblast in the extracellular matrix, we're going to get two enzymes to come in and cleave the C-terminal and N-terminal sides of this procollagen. So now that we've excised our procollagen out of the cell, we no longer have a need for these end pieces, which were the hydrogen disulfide bonding that occurred when we exocytosed it out of the cell. The reason we had to form those was to create the procollagen molecule that would stay together during export. So now that it's outside of the cell and in the extracellular matrix, we have no need for it. So essentially what happens is we get two enzymes to come in, which are N-collagen and C-collagen peptidases. Not an English major. What these peptidases do is they cleave those C termini and N termini ends of the procollagen, creating the monomer form and almost the final form of our collagen. And that's called tropocollagen. And now that we've released this tropocollagen, what's going to happen is the tropocollagen is actually going to stack. It's going to stack here on top of other tropocollagens, and we're going to start to create our collagen fiber. But in order to do that, we need to covalently cross-link, covalently attach these tropocollagen fibers together. And the enzyme that's going to do that is called lysine oxidase. And so lysine oxidase needs two things to function. It needs vitamin B6, which is also known as pyridoxal phosphate, and it needs a copper atom. And to explain a little bit more about these, because I didn't explain what the function of these things were, vitamins, for the most part, when interacting with an enzyme, are organic molecules called coenzymes. And they help to enhance the function of a certain enzyme doing its reaction. These metal ions like copper and iron are what we call cofactors, and they, along with coenzymes, can work together to enhance an enzyme's function. So again, lysine oxidase is the final enzyme in this synthesis pathway that will cross-link different lysine residues in the tropocollagen molecules that are stacked on top of each other to create this long, extensive collagen fiber. And that's what we call covalent cross-linking. So now we're just going to look at the steps one more time and kind of go over the synthesis pathway that collagen has. So the first step of the synthesis pathway is to translate and transcribe the mRNA transcript to create the polypeptide alpha chains, which are the pre-procollagen chains. Next, they'll be hydroxylated in the rough endoplasmic reticulum by proline and lysine hydroxylases. That's where we add hydroxyl groups to the proline and lysine residues. Again, that enzyme requires vitamin C and iron. If lacking, that will cause deficiencies, which we'll talk about in a second. Once the pre-procollagen has been hydroxylated, we can then glycosylate hydroxylysine residues to help form our procollagen molecule. And once those residues have been hydroxylated, we then get the assembly of the three procollagen chains to form the final procollagen molecule. And that occurs through covalent cross-linking with hydrogen and disulfide bonds at the edges of the procollagen molecule. The procollagen molecule then gets excreted outside of the fibroblast, where it interacts with N and C peptidases to cleave the ends of that procollagen molecule, turning it into tropocollagen. Tropocollagen then stacks on top of itself with other tropocollagen monomers to form the collagen fiber through covalent cross-linking. The enzyme that does that is lysine oxidase, and lysine oxidase requires vitamin B6, also known as pyridoxal phosphate, and copper to do its work. Deficiencies in any of those will cause certain disorders that we will look at in a second. So now let's look at some of the common diseases that are affected or that arise due to defective collagen synthesis. And the first one I want to take a look at is osteogenesis imperfecta. Osteogenesis imperfecta is an autosomal dominant disorder in the collagen 1A1 and 1A2 genes. And what happens is we get defective production of type 1 collagen. And if you remember from the different 4 types of collagen slide, type 1 collagen is responsible for bone formation. And so specifically within the synthesis pathway, when we get the 3 alpha chains and start to form our procollagen molecule, that process is defective in osteogenesis imperfecta. And so what we get is we get weak and brittle bones that can actually fracture in utero. And so a common diagnostic test that you'll see to see if a child is born with osteogenesis imperfecta is upon x-ray, they'll have broken bones either at birth or in utero. Another common symptom is blue sclera. That's one of the key features of osteogenesis imperfecta that you want to look at. So now that we've talked about osteogenesis imperfecta, let's look at another disorder that can arise in collagen synthesis. So scurvy is actually due to a deficiency in vitamin C. And a lot of the main stories around scurvy are seen in people or settlers traveling on boats. And they didn't have any fresh fruit or produce on their boats, so they would lack vitamin C. And you'd often hear stories of people bleeding from their gums, losing teeth, bleeding easily, and just very weird symptoms that in the early day and ages, nobody really knew the pathophysiology of. We now know that due to this vitamin C deficiency, we can no longer get effective hydroxylation of proline and lysine residues via hydroxylase enzymes, because vitamin C is a coenzyme for hydroxylase. And so that causes an issue in the formation of procollagen, which is going to have a lot of side effects, including these bleeding gums, loss of teeth, and issues with bone formation and stuff like that. The next disease we're going to talk about is Ehlers-Danlos syndrome, which arises from defects, from defects in the cleavage of the procollagen into tropocollagen and cross-linking of tropocollagen. And so what that's going to cause is it's going to cause an inability to form our collagen molecules, which is going to have issues and result in hyperextensible skin, hypermobile joints, and tendency to bleed. And if you think back to the types of collagen, type 3 collagen, which is deficient in Ehlers-Danlos syndrome, results in abnormal blood vessel formation and easy bleeding. And the final disease that we're going to talk about today is what's called Menkes disease. And in Menkes disease, we get a defect in Menkes protein. And Menkes protein is located in the gastrointestinal tract, and it's responsible for the absorption of copper. And if you remember, copper is an important cofactor in which enzyme? It's important in lysine oxidase. And lysine oxidase is the enzyme that's responsible for the covalent cross-linking of tropocollagen monomers into the collagen fibril. So if we get defective collagen fibril synthesis, we get brittle, kinky hair, growth retardation, and hypotonia. Those are the main symptoms of Menkes disease. I hope this video was helpful for you guys. Please let me know if you have any requests for future videos, and don't forget to like, comment, and subscribe. Thank you very much.